Hi everyone, my name's Tom and I'm going to be one of your demonstrators for plant physiology and ecophysiology this semester. We're here in the glasshouse, which is where many of your pracs are going to take place this semester. In the first few pracs, we're going to be looking at water relations in plants and plant adaptations to different lighting conditions. These pracs will be delivered in an online mode to you, and you'll be writing lab reports based on the data that are collected during these prac classes. In your remaining prac classes, you'll be forming groups and conducting an experiment where you'll be making comparisons amongst different groups of plants that you have grown yourselves. You may wish to use some of the methods that I'm going to demonstrate to you today during your experiment, so make sure you pay attention closely to these methods. So this prac is all about stomatal behaviour and xylem or stem water relations. Water is very important for plant life, and plants use huge amounts of water. As plants photosynthesize, carbon dioxide diffuses into the leaf, but water diffuses out of the leaf. Because the driving gradient for carbon dioxide is so much smaller than that for water loss, for every molecule of carbon dioxide gained by the plant loses about 400 molecules of water. Because of this, plants require huge amounts of water and have developed specialised adaptations to ensure that they use water efficiently. In one of the exercises of this prac, we're going to be looking at those water relations at the leaf-atmosphere boundary. And we're going to be measuring something called stomatal conductance. Stomatal conductance is a measurement of the rate of water loss over time. When we do these measurements, we have two groups of plants that we're going to use. We have some dwarf bean plants that have been grown in the glasshouse here. We have one set of plants that have been grown on the wet mat here. These have free uptake to water whenever they need it, so these are our well-watered plants. We also have a group of plants that have been grown on saucers. These do not have access to water from the wet mat, and they are watered manually. These have been watered in a way so that these are our water stress plants. So in this next exercise, we're going to be measuring stomatal conductance with something called a leaf barometer. This is this instrument I have in my hand and next to me here. This is a portable instrument, so you can measure the stomatal conductance on the plant when it's in its relevant environment. This is very important because a plant that's in a sunny condition like this is going to photosynthesize a lot more and lose more water in a dry environment than a plant that's kept in a shady condition. When using the leaf barometer, it's very important to make sure it's been turned on for about half an hour before you use it. This gives the instrument time to warm up so that the measurements you're getting are accurate. When you do use it yourself, make sure you don't turn it off when you're done, or else this will confound the measurements of the next group. When you do make the leaf barometer measurements, make sure you use a very steady hand, as this will cause the measurements to fluctuate if you wobble around. Clamp it onto the leaf surface and make sure that you have the leaf spread across the entire area of the leaf clamp. The measurements will begin to fluctuate when it's first adjusted, so keep it on there for a while and look at the measurements as they're coming out. As these start to stabilise, this is the number that you can record, and make sure you record the units as well. On each of the plants that you measure, not all of the leaves are going to be identical. It's therefore important to do multiple leaf barometer measurements on the same plant to make sure you get a stomatal conductance reading that is representative of the entire plant. An understanding of xylem hydraulic properties is very important for understanding the behaviour of plants. Hydraulic conductance is a measurement of the ease of which the water can move up the xylem of the plant, and is very important in understanding how water can be transported up the plant. This is very important because we know plants need to use water efficiently to photosynthesize and therefore grow. When we look at hydraulic conductance, it's that flow of water that occurs over time, and this is also dependent on the pressure gradient that's driving this flow. We can see that in this equation here. We have K, which is our hydraulic conductance, and this is equal to the flow rate divided by the pressure difference that's causing our water to flow. Our hydraulic conductance, therefore, is in the units of volume of water per time per pressure. With plants in the field, the pressure gradient that would cause water to flow would be the difference in water potential between that of the root and the leaf surface. In this experiment that we're going to be using today, we're going to use something called the Drippy Stick apparatus. What's going to be driving our pressure gradient is this water column here. Based on the height of this water column and gravity, it's going to cause a pressure gradient that will flow down through this hose, through a number of hoses here, and we will attach branches to the end of each of these hoses and measure the rate at which water flows through these branches. So we have branches from a lily-pilly plant that are going to go onto the end of our Drippy Stick apparatus. We're going to measure the rate of water flow through each of these branches. To prepare our branches, we need to have them submerged entirely underwater. We can start peeling off the leaves, and then we're going to cut off the ends of each of the branches. It's very important that this is done entirely underwater, because otherwise when we do start to cut off the ends of our branches, the water column inside the branch will recede into the branch, and we'll therefore get a break in our water column. Okay, so now that I've got it all cut up, I can bring it up out of the water, and I'm going to seal up the outside of those branches with some parafilm. This will ensure we don't have any water loss where those leaves were that were just taken off the plant. The easiest way to do this is to get a strip of parafilm like this and start wrapping it diagonally around the stem, so it should spiral around the plant. This only needs to be one layer thick, and it's just to ensure that we don't have any water loss there. Oops, we've got a tear there, but we can start again. Very importantly, we don't want to have any water flow between the parafilm and our plant stem, or else we will significantly overestimate the hydraulic conductance as we get more and more water flowing through. There we go, so just lightly putting a bit of tension on the parafilm as we wrap around. With any excess parafilm, you can just break that away there. Okay, with our sealed up branch, we're going to attach that to the end of our Drippy Stick apparatus. This should be wedged up there far enough so that there's no way any water can come down through the outsides. Once the water flows through our branch, we're going to collect it into this vessel here. I've pre-weighed this vessel so I know exactly how much it weighs. When I weigh it after a certain time period, I can therefore calculate the volume of water that I've collected that has come through our branch. So we have our tap on our big water tank at the top. The only thing left to do is turn on our tap that connects to our branch and start a stopwatch. There we go. So we're going to wait about 5 to 10 minutes and see how much water has dripped through into our collection vessel. So after 10 minutes has passed, we've stopped the time, removed our collection vessel, and we've then gone and weighed that vessel again, so we can calculate the volume of water that has flowed through our branch. We then need to measure some branch characteristics. Obviously, a branch with a wider cross-sectional area or a wider diameter is going to have more water flow through it. Also, a longer branch is going to have more resistance to water flow, so branch length is also going to impact the amount of water that flows through it. We can turn off our tap and remove our branch from the Drippy Stick, and we can remove some of the parafilm and a layer of bark. We can use our vernier calipers to measure the diameter of our Drippy Stick, and if we divide this by 2, we can work out our radius, and by using the area of a circle, pi r squared, we can work out the cross-sectional area that water flowed through. We also need to measure the length of our branch, and hydraulic conductance is commonly expressed per branch length. So if we look at that there, we've almost got a perfect 10 centimetre length of branch. The final thing we need to measure is the height of the water column. The height of the water column is what has driven the water to flow through our branch. This is dependent on gravity and the height. If we were to have this water column much higher, we would have a bigger pressure difference, and this would cause more water to flow through our branch. So we can measure the water column from the top of the water column all the way down to the top of our branch. With these variables, we can then follow the instructions in the lab manual to work out the hydraulic conductance of our lily branches. So after measuring the surface area and the mass of each of our leaves, we're going to store them for 5 minutes and then measure the change in weight over that period. We're going to store one of these leaves in light and one in a chamber that's completely sealed out or removed from all light. So I'll put one inside our chamber. And I'll leave the other one just here. Very importantly, start a stopwatch now and start timing. Just before, as I said, a larger leaf is going to lose more water. If we leave these leaves for a longer period of time, they're going to lose more water as well. So start a timer, and in 5 minutes we can come back and measure the mass of these leaves and look at the weight loss. That concludes these online practices where we've looked at water relations in plants, as well as plant responses to growing in light and shaded conditions. Make sure you use the data that's been generated from these practices for your lab reports. You can refer to your lab manual for detailed instructions on how to write these lab reports and what needs to be included in each lab report. You can also refer to these videos to see the methods that are available for you to use in your group projects. You might want to look at these videos to get an idea of what you can measure in the plants that you look at, as well as how to make these measurements. Best of luck with the rest of the semester.